Welcome everyone to the Biochemistry Colloquium. It's my pleasure today to introduce Professor Brian Smith from the Medical College of Wisconsin, where he's an associate professor of biochemistry. And Brian and I sort of go way back. I've known him since I was a grad student. When I was a grad student, I wanted to do these experiments involving rapid chemical quench kinetics, and Brian taught me how to use the instrument for this, and it was kind of a fun time, making these like little Atari sounds with this very old looking instrument. So we first connected over mechanistic enzymology, and Brian learned how to do this type of work in John Deneu's lab here at UW-Madison, where Brian was a chemistry PhD student and in the biotechnology training program. And in the Deneu lab, Brian did some really cool work developing chemical probes and doing mechanistic study of SIR2 family deacetylases, so important erasers of histone modifications. And then he went on to do a postdoc with Michael Marletta, which is where I met him at UC Berkeley. And then he also moved to Scripps there. And he did some cool work understanding the domain architecture of nitric oxide synthases and the role of nitric oxide in cell signaling. And now in his own lab, Brian's continuing along this like chemical biology vein, studying post-translational modifications and developing some cool chemical probes to study readers of acetylation marks or acylation marks in general on histones and their importance in driving transcription and assembling transcriptional complexes in cell signaling. So I guess in these times, I think community is really important. So it's cool to have someone local to come in and give the seminar. And I wanted to highlight a couple of awards that I think show what an important part of the community is. Brian is at the Medical College of Wisconsin. He's gotten numerous awards for graduate student education and also medical student education, and also awards for faculty service, so service to his colleagues in his department, which I think is really cool. And without further ado, I'll let Brian tell you about his exciting research.
Okay. Thanks, Amy. The microphone's carrying. Good. Okay. Yeah, it's great to be back. This I feel like is an important milestone in anyone's career when they're finally invited back to their oh, it's Siri. I probably hadn't do not disturb. To be invited back and be able to talk about your research and see all the faculty and that you've admired your entire career and hopefully have done yourself at least a little bit proud in their eyes. And just to meet the new faculty as well. So I purposely tried to target the faculty that I hadn't met before and who I requested because they're the people that I want to get to know. And it's been a great day. I've had the most students that I've ever had at a lunch. That was great and really had a great conversation there. So thanks again, Amy, for the invitation. So as Amy mentioned, we study epigenetic proteins. We focus mostly on the histone language, where we're focused on different post-translational modifications that happen to histone tails and how that affects chromatin architecture and compaction, and therefore gene transcription. And really this started in the Deneu lab, my interest in this with interest in lysine acetylation, where I did my PhD completely on sirtuins and enzymology and targeting domains, as Amy mentioned. But now everything I'm going to talk about today is on bromodomains, which we switched to these reader domains that read sites of lysine acetylation or acylation. And so I'll talk about this in a couple different contexts, but there's several different ways that bromodomains can affect transcription. One is often these bromodomains, these reader domains, are in the context of larger proteins, where they might have another writer or eraser domain that then puts on or takes off a neighboring post-translational modification. One example of that is TRIM33, which is a ubiquitin ligase, which I'll talk about a little bit later in the talk. Another are these chromatin remodeling complexes, one of which is the PBAF complex that contains a bromodomain containing protein called polybromo-1 that can either open or close chromatin, making the DNA either more or less accessible to be transcribed. And then they can also just have other protein-protein interaction domains like RNA, BRD4, which can recruit PTFB, which then can phosphorylate and recruit RNA polymerase II to then actually lead to transcription. So that's a few different ways that these bromodomains can affect gene transcription. And my lab is interested in or at least what I'm going to talk about today, is several different aspects of this. One is how can we improve selectivity of inhibitors to make better inhibitors that are more selective for the bromodomain targets that we're really interested in. And I'll talk a little bit about PBRM1 bromodomains in that context. Another is how variants, so disease-associated, either rare disease or cancer-associated variants, how that can affect protein function. And then talk a little bit about our work in type 1 diabetes, where we're looking at bromodomain inhibitors in this context and how we can be more selective for the cell types and the transcriptional targets that we're really interested in. And I'll talk a little bit about TRIM33 and that binding to not just acetylation, but other acylations like histone lactylation and how that can affect macrophage physiology. So the first family I'm going to talk about today is the PBRM1 family, which is over here on the right. But bromodomain family overall, we have 61 bromodomains and 46 proteins. So right there, you can see that there's several proteins that have more than one bromodomain. PBRM1 is unique though in that it has 6. So the most any other human protein has is 2 bromodomains, but PBRM1 has 6 different bromodomains. And it's a unique member. It's actually the P in the PBAF complex over here on the left. So we really wanted to develop inhibitors for this bromodomain containing protein. And one reason why, there's several reasons, but one reason is that if you look at humans or mice that have PBRM1 knocked out or a loss of function mutation, then those patients respond better to cancer immunotherapy like anti-PD-1 immune checkpoint therapy. So the idea here is if we take these patients and we look at the ones that aren't as responsive and then we inhibit PBRM1, can we make them more responsive to these cancer immunotherapies? And one reason why this might work is another lab, Waco Quee's lab, who's at Northwestern. He used to be our neighbor at in Diversity Blood Research Institute at the Medical College of Wisconsin, found that if you knock out PBRM1 and therefore knock out the PBAF complex, you get more effector T cells, which are the T cells types that you really want to kill the cancer cells, and less of the exhausted T cell type. So the idea would be if we can inhibit PBRM1 pharmacologically with a small molecule inhibitor, do we also get more effector T cells relative to the exhausted T cells? And so we don't want to do drug discovery in a vacuum. We want to make sure that we're not just reinventing the wheel. So first thing we always do is look, what's the existing state of the inhibitors? And when we started this project, all the inhibitors that existed, and there's a few that inhibited PBRM1, BD5, the 5th bromodomain really well. But all those bromodomain inhibitors also inhibited SMARCA2 and 4 with similar affinity. So these would be, in this context, off-target effects. So we put this as maybe more of a sledgehammer approach. And why is this important? It's mostly important because in the context of the 3 main BAF complexes, SMARCA2 and 4 are common to all 3 up here. You can see that all 3 have that SMARCA2 or 4 subunit. So if we inhibit SMARCA2 and 4, we're inhibiting all human BAF complexes, whereas we just want to inhibit PBAF. So if we can get PBRM1, we've also done a little bit of work with Emily Dijkhuizen on BRD7 inhibitors. If we could inhibit either of those, we'd be more selective for just the PBAF complex and not the entire BAF complex family. And so how do we do this? So another hat I wear is as a co-director of our program in chemical biology, which is just a collaborative research entity. One of the things that we've implemented is a robust fragment screening pipeline by protein detected NMR, where we can take, in this case, we take mixtures of 12 compounds, at least initially, to multiplex them, and then take N15 labeled proteins. Bromodomains work great at this because they're small. They're about 15 kilodaltons, a perfect size for protein detected NMR. Put these compounds in. We take a 2-dimensional NMR spectra where we have N15 on one axis, proton on the other, and if a small molecule binds, the nearby amino acid peaks will shift. That's indicative of binding. And then we use that information to then get the binders versus the non-binders in our screen. And so our initial version of this was on the cover of Chem Biochem, where we developed a few different bioinformatic or high throughput ways to look at the spectra, one of which we call difference intensity analysis, which I won't have time to talk about in detail. But then once we have the hits from the initial 12-plex, we parse that down into individual compounds. So for PBRM1, this looked like this, where at least for one library, we have 4 different libraries on campus. These are just commercial libraries that you can buy from Maybridge or Tarkemol or enamine or Life Chemicals. Those are the 4 that we have. Then we do principal component analysis to then separate control. So anything that's near the control here would be a non-hit, and then the ones that separate from control are the hits. The difference intensity analysis, the bigger bars are indicative of hits. And then we do 4 mixtures of 3, take those hits, individual compounds to get eventually our hits. In this case, we got 12 hits from this particular library. And then after that, we develop these compounds. So the first thing we do is we purchase them from another source so that we can, and then check to make sure the chemical matches the NMR spectra and everything checks out. And then we do titrations. And so from these titrations, we get both binding affinity, but also where these compounds are binding on the protein. So depending on which peaks are shifting the most, that's indicative of where that binding site is most likely to be. And this does distinguish it from other screening techniques where it's a little bit harder sometimes to tell between something that just non-specifically aggregates your protein versus something that actually is a true inhibitor. That's immediately obvious in an NMR spectrum if you just lose all your signal versus seeing clear shifts. So it helps us get rid of those false positives really quickly. And so this is one of our better hits that we've gotten from the screening pipeline with a KD value of 45 micromolar. So once we have that, we do pretty standard medicinal chemistry, changing different substituents. And in this case, we were able to optimize to something that bound to about 216 nanomolar and using our program in chemical biology synthetic chemistry facility. So this is something I was talking to the students at lunch, is that my skills in chemistry probably aren't unique. I see chair of chemistry in the back, Dr. Gelman. I don't have particularly unique chemistry skills, but put in a medical school environment where I'm the only chemist, I'm suddenly super unique and super valued. So I get to collaborate with everybody on anything that's chemistry because I'm the chemist, which sometimes is daunting, sometimes is really fun, but it's really helped us grow. And that's why we publish a lot of collaborative papers. If you look at my papers, they're pretty much all collaborative. And we've collaborated with, I think, 80 different faculty and probably almost 20 different departments on campus. So it's been fun, but I would like to hire more chemists. So if there are people that want to stay in Wisconsin and think they could contribute in a chemical biology or medicinal chemistry, hopefully we'll have a way to hire you in the future. I just said the impact already, so I'll just skip ahead. But with a relatively modest investment of $5 million, we've been able to be a part of almost $60 million in competitive or external grant funding. So I think, to me, that's a pretty good return on investment. Okay, so we have an initial compound we developed, but the whole point of this was to get something that's selective for SMARCA2 and 4 over the PBRM1 bromodomains. And so we did this in several different ways. Isothermal titration calorimetry. I'm only going to show you our different scanning calorimetry data, where if a small molecule binds to the protein, that makes it more thermostable, and that's indicative of binding. And so we took one of the previous non-selective chemicals or compounds and showed that, yes, that stabilizes SMARCA2 and 4 bromodomains in addition to the PBRM1 bromodomains. But importantly, our compound only stabilized PBRM1 BD2, which is what we screened against, as well as a little bit BD5, but did not stabilize 2 or 4 at all. And we also saw the same thing in ITC, that our compound does not bind the SMARCA bromodomains at all. So we got our selectivity, so that was great. But we wanted to understand why it was selective, because it wasn't really immediately obvious, given that the compounds were similar. So Ray, a senior grad student in the lab, saw the crystal structure of this bound, and we used that to then get an insight to why our compound was selected. And so what we did is we used that structure, looked at residues that were different in SMARCA versus PBRM1 that were nearby, near or making contact with our inhibitor. We also used our NMR data, so we knew which amino acids shifted when we added a small molecule, and correlated all the data that come down with, I'm going to show 4, but we ultimately made 5 different mutants that were different in SMARCA versus PBRM1. And thankfully, all the mutants that we looked at were the same in SMARCA2 and 4, so we only needed to make one set of mutants in PBRM1 in each residue. So what we found is relative to our wild type Katie, this is a Katie from ITC, mutating this leucine was a modest effect, this tyrosine a modest, relatively modest, and this valine, but the biggest effect we saw was mutating this tyrosine 217 to leucine. And then we did the inverse experiment when we took SMARCA 2 and 4 and then added that, changed that leucine back to the tyrosine and saw from what we had, it was zero binding, just a flat ITC curve. We gained binding back into the SMARCA 2 and 4. So we think this leucine to tyrosine switch is a key element. And if you look at this in terms of our modeling, that tyrosine is kind of flipped out in the PBRM1, but that leucine sticks towards this methyl group in our compound. And so we think there's a steric clash here that's introduced once we have that leucine in the pocket. So we're using that information to help design more selective inhibitors and take maybe some of the unselective inhibitors and make them selective, because the unselective inhibitors have the advantage of potency. We have the advantage of selectivity. We'd like to marry those both together. So another thing that's great about grad school is you meet other people that you collaborate with way later in ways that you didn't expect. So Emily Dijkhuizen was a graduate student in Laura Kiesing's lab. We weren't working at all the same things when we were grad students, but we slowly have moved, our research programs have moved towards together to the point that we've collaborated on several papers. One of which is this paper where she took the LN-cap cell line, which is sensitive to PBRM1 knockdown, and then also looked at the PC3 and the RWP1 cell lines that are insensitive. So we thought we could use this as a readout for on-target selectivity versus any sort of off-target, just general toxicity of our compounds. So we took 3 of our compounds, one of which is that initial unselective compound, and 2 of our compounds that we were more selective, looked at that. And indeed, we see loss of viability in the LN-cap in all 3, consistent with inhibition of PBRM1, but no inhibition of, or no loss of cell viability in the cell lines that shouldn't be sensitive to PBRM1 knockdown. We do see a little bit of inhibition of viability or loss of viability with this PC3 with the unselective compound, further showing that we think that our compounds are more selective. And then the IC50 for this effect was about 9 micromolar. So we're in an okay spot. Ideally, we'd like to get this into the low into the nanomolar range. So right now, what we need to do is use our structure to do some structure-based drug design to hopefully get that potency, increase that potency a little bit before I would be comfortable calling this a good probe compound. So a couple other things that we wanted to do to make sure that we're hitting on target is one, that we compared our inhibitor to PBRM1 to a BRD7 inhibitor that Emily Dijkhuizen that had developed. And we saw, since these are the 2 of the unique subunits in PBAF, we'd expect roughly similar transcriptional changes. And indeed, we see that just looking at RNA-seq, the changes that we see largely overlap, consistent with it being a similar mechanism of inhibition. Another thing that came out of this is that in this case, we're looking at prostate cancer cell lines, and enzalutamide is a standard of care for prostate cancer. So we looked at the transcriptional changes we see with enzalutamide versus our inhibitor. And what we see is very similar transcriptional changes with our inhibitor of PBRM1 as we do with enzalutamide. But this is a completely different mechanism. So what we're doing right now is starting to look at enzalutamide-resistant prostate cancer cell lines to see if our inhibitors still have activity against those enzalutamide-resistant prostate cancer cell lines. So hopefully that would be a way to treat enzalutamide-resistant prostate cancer. Okay, so that's the first story on the bronamine inhibitors. I want to move now to our PBRM1 variants and how we're looking at those in the context of cancer therapy. And this is part of a larger group of researchers in the Mello Center for Genomic Science and Precision Medicine, where another hat I wear is the director or co-director of our structural genomics unit, where essentially we work with other groups in the Mello Center. One group does more computational modeling of variants that we get either for rare diseases or cancer from sequencing or from online databases. And then we do all the protein biochemistry. So like circular dichromosome, enzyme kinetics, focus mostly on histone binding domains and modifying enzymes, and then another group does the cellular assays. So what's nice about it is I get to do what I really like to do, is a lot of protein biochemistry work, but it fits much nicely into a whole clinical pipeline. So the translational aspect of it is high in doing those types of assays that I really like. So we're looking at missense variants. So this is going back to that same slide where I said if we can inhibit PBRM1, but then the question is, what about all these people that have missense variants? This is all loss of function mutations where your protein's completely gone. What if you have a missense variant? Are you closer to the yellow here? In which case, you're maybe not going to respond as good as well to anti-PD-1 immune checkpoint therapy, or you're closer to the blue where you'd be a high responder, or somewhere in between. I think that's valuable to know because that might dictate which treatment course you might take for your cancer. So looking at this is a little bit daunting. There's almost 3,000 mutations that have been cataloged for cancer in PBRM1. And about a third of those are missense mutations. About half of those are in the Brown domain. So there's about 400 or so missense mutations that we could look at. So we needed a faster way than making these one by one and looking at them in vitro. So we developed a computational pipeline where we take two-dimensional sequence information at one level. And this is pretty much where the clinical pipelines are right now. They're pretty, I don't want to say archaic, but like they could go further to look at sequence and try to get a sense of what's the likelihood that that particular mutation is deleterious or pathogenic. But what we're doing is then taking that same information, putting in the context of the three-dimensional structure of the protein, and also doing molecular dynamic simulations on those variants to get an overall sense of how likely a certain variant is to be deleterious to its function and give it a score between 0 and 1. So that eventually looks something like this, where something closer to 0 is more likely to be benign, something closer to 1 is more likely to be deleterious, and then we can further categorize those by the potential mechanisms of action. So we do this, and then we take a few of those and try to validate which one, how good our predictions are. And we can do that by simple things like just thermal stability of it, overall protein fold, circular dichroism for overall folding of the protein. We can also do it different ligand binding assays or enzyme kinetic assays, and we can also do cellular assays. So this is kind of in the order of throughput. The melting temperature is very high throughput, ligand binding fairly high throughput, but the cellular assay is fairly low throughput. So we tend to triage things as we go. And so one particular cancer that this is really important in is clear cell renal cell carcinoma, which PBRM1 is the second most mutated gene. So that we looked at, for this next level of study validations, we purposely selected ones that were CCRCC, or clear cell renal cell carcinoma, just to narrow our pool of variants that we could look at. And then we also compared those to population variants. So these are variants that just naturally occur in the population. We have no real reason to think that they're deleterious at all to the protein function. And then we score both of those. And ideally what you'd see is that the ones that are population should be more likely to be benign. The ones that are cancer associated are more likely to be deleterious. And we see in our integrated pipeline a pretty good scoring of the population, where we classify those mostly as benign. And then the ones that are in the CCRCC, mostly deleterious. The CAP is a clinical annotation of pathogenicity. That's pretty much what typically used, I think, in the clinic right now. And then alpha missense is using AlphaFold, and then to determine how much, if the missense variant would be deleterious or not. And both the clinical annotation of pathogenicity and alpha missense, I think, tend to misclassify the population as deleterious, whereas we do better at that. And then the CCRCC variants, similar, maybe alpha missense does a slightly better job, but I think it tends to just have a bias towards being more likely to be pathogenic. And so does this have any basis in reality? If we take one version of our score, our structural pathogenicity score, and then compare that to the actual experimental thermal stability of the protein, then we see a fairly good correlation. Some that are notably off this line, one of which is this N106K. And so this is a unique variant in that it's at this really important asparagine. This is the asparagine that hydrogen bonds to the acetyllysine. So you would think that just maybe that wouldn't be captured in structural stability, but would be captured maybe in molecular dynamic simulations with the peptide, but is a slightly different mechanism rather than just overall unfolding of the protein. So right now we're working on how do we capture these ones that maybe have different mechanisms from our approach. Another interesting correlation that we saw, at least I thought was interesting and surprising, is when we looked at the overall stability of the protein. So this is a cyclohexamide pulse chase where we inhibit protein synthesis, and then we just look at how long it takes to lose the protein, how stable the protein is over time. And then when we compared that to our melting temperature, so I at least thought this was just surprising. So what we're looking at is an isolated domain, not the full-length protein, just one domain in the context of all the 6 barodomains that is less thermostable, and that correlated fairly well with the stability of the entire full-length protein in the context of an intact cell. To me, that was a little surprising that it correlated well at all. I would have thought it would be less correlated, so that suggests that we could maybe use the melting temperature somewhat as a proxy of the overall stability of the protein in the cell. Okay, so that gives you a rough sense of how we're approaching the disease-associated variants. So next I want to give us a little story about how we're looking at BET inhibitors and type 1 diabetes. So this in particular is looking at the BET family of barodomains. So this is another family of those tandem barodomain containing proteins. So I'll get to it in a second. So this is at the top of that tree right there. So this is a family of BRD2, 3, and 4. I'm mostly just going to talk about, and T, sorry, BRD2, 3, 4, and T. I'm mostly going to talk about just 2, 3, and 4. T is testis specific, so it's not present in the cell types that we're looking at here. So we largely ignore that in the context of our studies. But the nice thing here, there's been several what we call pan-BET inhibitors that have been developed. These pan-BET inhibitors inhibit both barodomains of all 4 of these proteins, including T. And so they're fairly non-specific with this within the family, but specific in the context of the barodomains overall. And so what inspired us is a study where they looked at the non-obese diabetic mouse model of autoimmune diabetes and did different, this isn't our work, but one lab, Diane Mathis's lab, and then Peter Thompson, treated for different 1-week windows, showed that if you treat these, then you delay the onset of type 1 diabetes or autoimmune diabetes in these mice. And this is shown in the same study here, just the axes, how they drew the axes are inverted. So this is interesting to us, but we really weren't sure about the mechanism. This paper, the eLife paper, largely attributed this effect to antagonizing NF-kappa B signaling, both in macrophages and beta cells. So that's where we started. And just in general, one way to think of type 1 diabetes is we have some sort of insult that leads to activation of pro-resident macrophages that can recruit T cells and B cells as immune cells through signaling through IL-1 beta. And then eventually that leads to reduced function, reduced proliferation, increased apoptosis, or just increased cell death of the beta cells. And if we can inhibit these processes through pan-BET inhibitors, potentially that's a way to delay the progression of type 1 diabetes. And so looking at this, what we think is going on, this is definitely not the whole story, but at least what we've looked at so far is in the context of macrophages, we think these BET inhibitors are decreasing NF-kappa B signaling, leading to increased inflammatory gene transcription, or decreased inflammatory gene transcription, and decrease IL-1 beta, which then leads to decreased IL-1 beta signaling within the beta cell, which also goes through NF-kappa B. And one thing that happened is the SIRT1 protein, so this is a sirtuin, those go way up. That's thought to be protective for a beta cell. We also see loss of NF-kappa B signaling. We also see decreased c-MYC, which is why these inhibitors are important in cancer. They decrease c-MYC protein levels, which decreases proliferation in the context of a cancer cell. The context of a beta cell, decreasing c-MYC typically results in an increase in beta cell and identity and function. genes. So that might all be another way that these are protective. So I'm mostly going to focus on, well, I'm only going to focus on the NF-kappa B signaling today, though. So we published one paper where we show that these BET-bronamine inhibitors seem to be acting in the nucleus, potentially inhibiting an interaction between the first bronamine of BRD4 and the acetylated P65 subunit of NF-kappa B. Breaking that interaction leads to, with a BET inhibitor, leads to less of these other factors that activate transcription of pro-inflammatory genes like inducible nitric oxide synthase. You get less nitric oxide, and that, since nitric oxide decreases cell viability and mitochondrial function, inhibiting this process results in increased viability and increased mitochondrial function. But one thing that bugged us about this story is that we knew that not all NF-kappa B genes are the same, and also inhibiting NF-kappa B itself in multiple stories. Previously, it showed that it's actually not helpful in the context of preventing type 1 diabetes. So the details of this aren't super important, other than this black line is activating or inhibiting NF-kappa B signaling, and that actually makes the diabetes worse, or a higher prevalence of diabetes. This other story has another inducible version of that inhibits the NF-kappa B signaling pathway, and that seemed to have no effect on the number of diabetic mice. So clearly inhibiting the NF-kappa B pathway broadly is not helpful, because all NF-kappa B targets are affected. Our BET inhibitors doing something that's more selective by only inhibiting a subset of the NF-kappa B targets. And so what we did there is we did a bulk RNA sequencing study where we looked at all the IL-1 beta induced targets, and then just put that on a list and just got all the NF-kappa B targets from that list. So there's a lot of other changes that happened, but we just looked at the NF-kappa B targets, and then we looked at how those were inhibited or changed by addition of the pan-BET inhibitor, BET-bronamine inhibitor, JQ1. And you can see here, some of them are decreased significantly, some actually are increased, and some don't change at all. So this clearly isn't just inhibiting all NF-kappa B signaling. So we see, here's some examples. Here's a gene that's significantly decreased by treatment with the pan-BET inhibitor. Here's one that's not changed at all. Here's one that's actually increased. So then we took all of these, integrated the area into the curve, and then classified them as either BET-induced, BET-insensitive, or BET-sensitive, or BET inhibitor-sensitive genes. And so from this list, we get about 22 that are insensitive, less than a 0.5-fold change, either up or down, and 30 that are sensitive and 2 that are actually induced. So what does this actually mean? So we looked at this, and I'm not going to show you all the details, but essentially when we looked at the pathways and the genes that were being affected, and we found something that I thought was really interesting. Basically that the genes that are sensitive typically are the ones that are doing extracellular signaling, something or at the membrane, the chemokines, the cytokines that then are recruiting the T cells or the other immune cells, whereas the ones that are insensitive are the ones that regulate the NF-kappa B pathway and also regulate the antiviral response, so all the internal signaling. And this is actually, I think, what you would want to do in the context of diabetes: decrease, maybe dampen down some of that extracellular recruitment of the immune cells that are killing or leading to loss of beta cell function, but keeping NF-kappa B signaling going, because you want that to self-regulate itself, because there's a lot of feedback loops that you want to keep intact. And you also would like to maintain an antiviral response, because virus infection is one of the things that can induce autoimmune diabetes. So what we're doing now is looking at that a little bit more in detail, not just the genes that are NF-kappa B dependent, but also all the other genes that are changed with BET inhibition upon stimulation with IL-1 beta. So this is great. We should all be taking, let's just put BET-bronamine inhibitors in the water, and then we can just cure all the diseases, right? So definitely not the case. So there's been a lot of clinical trials, mostly on the context of anti-cancer indications, either blood or solid tumors. And a lot of those trials are being halted due to some sort of dose-limiting toxicity. And we think this is really because we're inhibiting all these bronamines, we're inhibiting too much. We're inhibiting all 8 of these bronamines. It's leading to too much change in transcription across the whole body because these are ubiquitously expressed proteins except for BRDT. So you're affecting transcription all over the body. And especially if you think of what's our potential patient population for a type 1 diabetic, often that might be likely a kid that doesn't actually have they don't have type 1 diabetes, they're at risk. They might have some autoantibodies that make them at risk of developing type 1 diabetes. They might need to take this inhibitor for a while, although maybe just for a week based on the mouse studies. And so our risk tolerance is much higher or lower, I guess, depending on how you think about it. Somebody that has cancer, definitely wants to get rid of the cancer. Someone that is at risk of developing type 1 diabetes, which is a manageable disease, we need to have something that's a little bit safer. So what we're really doing now is trying to see what do we actually want to inhibit and how can we be more selective. And so just to emphasize the point, when we put a pan-BET inhibitor on a beta cell line, we see about 1,500 genes change. So this is a pretty high percentage of the transcribed genome. Imagine that happening in every cell type in your body. So what's been happening recently and since 2020 is a lot of labs have now started to develop more selective inhibitors. So we, for the most part, just use what other people have developed rather than develop our own here. So GSK and AbbVie have some BD1 and BD2 selective inhibitors. So this would be selective on this dimension here. And then we also have some BRD2 and BRD4 selective inhibitors, at least as reported in the literature, that would be selective in this gene dimension. So we're using those to try to narrow down what do we actually want to inhibit. And so at least in the context of a beta cell or islets, we seem to get the same response, at least for the genes that we've looked at so far, with a pan-BET inhibitor and a BD1 selective inhibitor. So we really only need to inhibit, whereas a BD2 selective inhibitor doesn't do anything relative to control. So at least in this case, in beta cells, we just need to inhibit the first bronamine, not the second bronamine. What about in a macrophage? So in a macrophage, it's actually a little bit different. A BD1 inhibitor that inhibits the first bronamine has an intermediate effect. Again, BD2 doesn't seem to have any effect, but that's not as much as the pan-BET inhibitor. And if we add a BD1 and a BD2 inhibitor together, then we reconstitute the effect of a pan and BET inhibitor. So if we just gave a BD1 inhibitor, we'd have a full response in beta cells and only a partial response in the macrophages. What about genetically? So in a beta cell, based on this nanobread experiment, the details aren't super important other than from this experiment, we can show that it seems to be BRD4 bronamines that are interacting with P65 in this context. So in a beta cell, we probably likely want to inhibit BD4 or BRD4. This is preliminary data, so it's not done, but at least it's consistent. And my student has repeated this, but he just didn't send me the data in time to put up here. But we have, in this case, this is in a raw macros, which is a raw 264.7, which is a macrophage cell line. In this case, it seems to be BRD2 that's important. So not BRD4, BRD2. And then we look in the context of a beta cell line versus a macrophage cell line and look at these BRD2 and BRD4 inhibitors, we see the exact opposite effect. So these are both supposed to be NF-kappa B driven genes, although from a different stimulus. In the macrophage, it's lipopolysaccharide in this case. In the beta cell line, it's interleukin-1 beta. It seems to be the exact opposite, even though they're both NF-kappa B driven genes. So there's something interesting going on there between macrophages and beta cells, and that's what Paul will be looking at. So the other dimension of selectivity that we have is targeting of the beta cell. So how can we selectively target an inhibitor to the beta cell? There's several different approaches you can do. The first one we've tried was taking advantage of the fact that beta cells are rich in zinc because they're the insulin-containing zinc granules. And so a lot of other labs have used this to put zinc chelators on their compounds. Groups that then are supposed to selectively lead to either selective retention or uptake of inhibitors. Short version of this is this doesn't work very well. So that's why I'm not showing you the data. but we were at least able to get paper out of it. So right now we're looking at other approaches where maybe antibody drug conjugates might be a smart way to go, looking at for cell proteins that are selective for the surface of the beta cells, and then conjugating that to the bronamine inhibitors. So we can get selectivity by inhibiting BD1 and 2 selectively, maybe inhibiting BRD2, 3, or 4 selectively, and then maybe targeting those inhibitors selectively to the beta cell or the macrophage. Okay, so now I want to talk a little bit about another macrophage story, but from a completely different angle, where we're really interested in how metabolism can affect gene transcription. In my case, I'm really defining this as metabolism leading to different acyl-CoA pools, which then leads to differences in either nucleosome or other protein acylation. And then how is that interpreted by bronamines? Is acetylation the same as all the other acylations or not? And if we look at this, this is mostly Yingming Zhao's lab at the University of Chicago, where we've seen in the last 15 years or so, almost 20 years or so, that there's actually a wide variety of other acylations that happen on histones, on nucleosomes, beyond just acetylation. And so a lot of these are metabolically derived in different ways. The one that we're going to focus on today is lactylation, which is derived from lactate. So why is this interesting? So this is a lab paper by Lev Becker and Yingming Zhao, University of Chicago, where they looked at when you have an increase in lactate after stimulating macrophages, in this case with LPS and interferon gamma, see an increase in lactate and then an increase in histone lactylation. That increase in histone lactylation correlates with basically a change from inflammatory gene transcription to anti-inflammatory or pro-reparative macrophage gene transcription. So one common marker for that is inflammatory gene is inducible nitric oxide synthase, and then arginase 1, which degrades arginine that's needed for the nitric oxide synthase to work, then comes on later. And these genes seem to correlate with where that these H3K18 lactylation sits on promoters. So our question here was, okay, we get histone lactylation, well, we get glucose, we get glycolysis, we get lactate, that leads to increased histone lactylation. Then what? What's interpreted after that? So the first step we thought to look at was, do bronamines bind to histone lactylation? So if 61 bronamines, lactylation is not that different than acetylation. Maybe some of these bronamines can bind. So we did a screen by a technique called alpha screen. Details aren't super important, but here's a diagram of how it works. And we had different histone peptides. So these were ones that were found in actual proteomics from Yingming Zhao's lab. Then we took all the bronamines that we've expressed and purified in the lab and then saw which one increased signal. And we got one really clear hit in TRIM33B. And so we're excited about that. Made it easy to know which one we should follow up on. And so one way we followed up on it is just to do an NMR titration. We also looked at it by isothermal titration calorimetry, and we saw clear binding of the lactylation that correlated with about the same affinity we get for acetylation. Then Sarah did a pull down just to show that we do see this binding in cells. So the important part is this bottom blot, where if we have lactate and TRIM33 present, both with an HA tag, we pull down histone lactylation, whereas if we don't have lactate or the TRIM33 present, we don't. So this seems to be true, at least in cell lysates as well. So one thing we like to look at, right, is why is this one selected? So in this case, we just did another sequence alignment. Again, used our NMR shift, chemical shifts to decide which residues we should be looking at, as well as our model. When we model this in, we noticed that there's this one residue here, E981, that's unique to TRIM33, not in even the most closely related one, which is TRIM24, which has an alanine at a position. And that seems to form a unique hydrogen bond to the lactylation, at least in our models. And this is just a model, not a crystal structure, but it's based on a crystal structure where acetylation was bound to TRIM33. So we did similar to what we did with the PBRM1 inhibitors. We took this glutamate, mutated to alanine. There we see loss of binding to lactylation, but not acetylation. And then we did the same thing where we took TRIM24, mutated that alanine to glutamate. We see increase or a strengthening of binding towards lactylation, but not acetylation. So that this seems to be an important switching residue for the ability to bind to lactylation or not. And it's actually interesting that it's in the paper, but evolutionarily, it shows up right around the time that you get essentially the innate immune system and an immune system develops macrophages. That's when that residue shows up. I don't know if that means anything, but at least evolutionarily, it makes sense when that residue shows up in terms of the function of the protein. Okay, so why is this actually exciting for macrophage biology? So another lab about 10 years ago knocked out TRIM33 and showed in bone marrow derived macrophages that when you knock out TRIM33, you just get a prolonging of this pro-inflammatory gene transcription, in this case, looking at interferon beta. And so looking at this time course here, 6 to 24 hours, that's the exact same time course that you see that increase in the histone lactylation. So it makes almost too good a sense. I was maybe too excited when I saw this, that it correlates exactly. When you see histone lactylation, that's when TRIM33 is important. So I can't tell you if that's really what's happening yet. That's Paul's job in his thesis, and he's working hard on that. But we think that that might be an important switch to control which genes go. So you have metabolism, you have a metabolic switch towards glycolysis, increased lactate, later on in macrophage activation, get a buildup in histone lactylation that then is interpreted by TRIM33 to then help switch from that pro-inflammatory to that pro-reparative or anti-inflammatory, however you want to define it, gene transcription. Okay. So I wanted to get to one last thing. Scott's in the crowd, right? All right, this is for you, Scott. I had to add this because of our conversation this morning. So I was excited. We just got this published. It was a long time coming, but Phil Cole gave us an editor's pick, so I was pretty excited about that. So in this case, we're looking at these tandem bronamains. And remember, I said we have quite a few tandem bronamains in our genome. There's about 10 proteins that have tandem bronamains. And so why two? Why do we need two for the function? And so at least at the most basic level on chromatin, you can imagine three potential interactions. You can have intratail, both on one tail, intertail, so two different tails, but within the same nucleosome, and then internucleosome, which could lead to chromatin looping. Now, I think the most interesting possibility is this chromatin looping if BRD4 is helping loop chromatin with enhancers or promoters. That to me is the most important one. So that's the one I'm going to talk about, although we did look at the other possibilities in this paper. So what we did was fairly simple. We expressed the tandem bronamains, purified them, then did small angle X-ray scattering to get a radius of gyration and an overall rough sense of the size and shape of the protein, and then whether it's ordered or disordered. And so from that, we didn't use Rosetta. This was before AlphaFold, so we were using Rosetta to just model the linker. So we took crystal structures of the two bronamains, modeled the linker, and then had Rosetta floppy tail generate a bunch of models, and then use the Sachs constraints to constrain those models either by the radius of, the radius of gyration here, or the maximum diameter. And then we only took the models that fit within those constraints. And then in this bottom one, we were looking at the interbroname binding site distances. So what we got from this is no clear energetic minimum between all these different sites, but we could have bronamains that are anywhere from about 15 angstroms apart. This is just looking at that key asparagine, how far they are apart from each other in BD1 versus BD2, all the way out to 175 angstroms. So this is a model of these two models here are either the furthest apart or the closest of our models. So it seems pretty flexible, and it's borne out by the Sachs data and the modeling. So if it's flexible, then it should be able to bind multiple nucleosomes, we would think. And so we looked at that two different ways, one of which is we looked at mononucleosomes. So these are purified calf thymus mononucleosomes that just have kind of a mixture of acetylation. We weren't really sure what acetylation we should use, so we just let the calf thymus decide for us. And then we put that in with our tandem bronamains. These are on a sucrose gradient, so the bigger they are, the more they'll migrate down the gradient. And so these, when we add the tandem bronamains, we see an increase in our migration towards something that's consistent with a polynucleosome size. If we just have the bronamine by itself, then it stays at the top of the gradient. Then importantly, if we mutate either of those important asparagines, so these are the ones that are binding to acetylation, then we see no shift relative to the control. It migrates essentially the same size as a mononucleosome. So this was consistent with its ability to basically scaffold multiple nucleosomes together. The reason this took a while to publish is we needed to figure out a different way to do that. And so I challenged Karina in the lab to figure that out. And she came up with this assay where we just look as the alpha screen assay where we, in this case, we use the same streptavidin bead on both sides, whereas normally you use the streptavidin bead to bind like a biotinylated histone on one side and then maybe a histag bead on the other side so that we have a nucleosome on both sides. And if we have a tandem bronamine, that should bring the two bronamines together. Eventually, once we saturate the sites, then we should lose the ability to scaffold because now they're monovalently saturated. And so that's what some people call the hook effect. So we saw that with BRD4 and BRD2, 3, and T, clear binding with a hook effect. And then if we mutate those individual bronamines, then we lose this whole alpha screening scaffolding. So from this, it's clear that we could do the scaffolding. And then we wanted to look at this a little bit bioinformatically to the extent that we could. So we looked at chromatin compartmentalization. This is often measured with high C. And this gives us a contact map. So basically, is this piece of chromatin close to this other piece of chromatin, regardless of how close it is in linear sequence of the DNA? Then we looked at which ones were transcriptionally active domains versus inactive. BRD4 correlates with transcriptionally active domains. And then we took all that information, then looked at what histone modifications were at those regions that also had BRD4, and we get a correlation with acetylation, unsurprisingly, not with methylation. So bronamines bind to acetylation, not methylation, as far as we know. So it makes sense. But what I thought was most interesting was when then we looked at where those correlations happen, those were strongest between the acetylation modification and BRD4 when we then separated those based on the annotation of is it a promoter, is a promoter flanking, or is it an enhancer or open chromatin? And by far the highest correlation was with certain sites of acetylation, only at promoters, but not enhancers. So that was the part that was surprising to me. I thought the BRD4 would be important for looping enhancers to promoters, but this suggests maybe it's much more important for looping promoters together and maybe making what people call transcriptional factories, where you have a lot of similar promoters that need to be transcribed at the same time near each other. All right, and so this is our general model that we have inactive, active chromatin compartments. BRD4 can maybe loop these promoters together to enhance transcription. Okay, so with that, I definitely want to thank all the people. I tried to thank them as we went, but I have a really good lab that everybody gets along. It's great right now. And also thank my collaborators, Brian Volkman and Francis Peterson, Rob Keys in program chemical biology, Israel Rudia, Michael Zimmerman in the structural genomics unit. All the work on beta cells is a collaboration with John Corbett, and then Emily Dykeisen on the clear cell or the prostate cancer cell lines and funding. And thanks everyone for coming. Happy to take any questions.
Do we have questions for Brian? I can, oh.
So in the section of the talk about the missense variance analysis, I think it a plot of your predicted 3D structural versus like melting temperature or something. I was curious how the data using your structural methods compares with the data from alpha missense, if you've ever compared the two.
We just started doing that, so we haven't compared that yet. That's definitely something we want to see, is really how ours compare, or do we need to be using alpha missense in the context of ours as one of the, we probably should just be using it as one of the many, because we use a lot of different tools and we're kind of just picking the ones that seem to not correlate with each other, so we're not just getting redundant information, but give us the most information with the least number of tools. But we haven't done that exact comparison, but that's a good one.
sort of just following up on that, have you, so this like MD method of figuring out what's going to have an impact seems really cool. And I was wondering, like, I've seen some people are coming out with these like LLM type models like EVO and EVO2 that claim to predict every mutation that causes human disease. Have you like compared it all with that to see get out of that line?
Yeah, the short answer, we haven't, but we should. I mean, we need to stay with the times. I guess everybody needs to do an LLM now. Yeah, this work was all done pre-LLM, well, not pre-LLMs, but pre-LLM boom. But we haven't looked at that in detail, but certainly keeping an eye on that and making sure that we don't fall off behind the times there.
I'm just curious, following up on that topic, how was it publishing a paper where you did work prior to AlphaFold with modeling structure? You know, did you get feedback, people pushing back against that since you're publishing it after the advent of structural prediction?
No, we didn't. I was a little bit surprised. I was worried. I mean, that paper took a long time. I mean, I think we initially put the preprint on there probably like 6 years ago. And so it took a while for us to get the right match of journal and data in there. But yeah, there wasn't much pushback, surprisingly, on that part of it. I would have thought there would have been more. But yeah, it's kind of weird when you think about like timing, making sure you're getting things out before the next greatest thing comes that makes sure what you just did completely irrelevant. But either one.
I have the mic, so I'll go first. Is there any way in your high throughput NMR screen that you can incorporate selectivity for a specific domain? Because it seems like you were able to identify a high affinity fragment and then do a bunch of medchem. And did it just happen to be selective for the PBRM1, or is that something?
Yeah, let me just guess roughly where we're at. Oh, almost. So this case, I forgot to mention this, so thanks for the question. Sorry, I hate it when people do this and just go backwards. Okay, here we go. So in this case, the way we designed it in, at least our thought was all the non-selective inhibitors were developed against the fifth row domain. And so we were looking a little bit about this. And so we looked at, so this is taking advantage of Emily Dykhuizen's work. She had mutated each of those prodomains individually. She found that 2, 4, and 5 are most important. So we took the one that was evolutionarily farthest away, at least in this tree, which is 2, and then did our screen against 2, thinking that screening that would maybe be slightly more likely to give us selective inhibitors. Also, 2 also had NMR assignments already developed, so we didn't have to do that part of the project, which was nice. So that's kind of how we built it in. But yeah, you're right. That's not always, the system's not always set up in your favor that way, and then you just have to counter screen. Hannah, I think, and then John.
The first isn't a question, it's a comment, but if your data for the variant effects are public, there's a database that I'm happy to follow up with you. It's called Protein Gym. And so if you want to upload your data there, then the LLM people can fight over it themselves.
Even better.
Even better, yeah. And my second is, again, just with your last story. I'm so interested because what made you decide to use Rosetta modeling for linkers? Because it seems like if it were me, I might think to first start with MD to be looking at the
probably, I mean, the honest answer is I didn't decide to do that. My student decided to do that, and I didn't argue with him. But yeah, I think there's lots of ways we could have done the linker. I think in this case, the answer is no matter what we would have done, is it's just a floppy linker. So I don't think it really mattered. We just had to do something to convince ourselves that it's probably floppy more than just, hey guys, trust us, it's floppy. But yeah, most people assume that it's floppy already. That probably wasn't the most interesting. The more interesting was doing the modeling. and constraining it to the SACS data and seeing how far apart the distance constraints were between the two pronomains, I thought. Yeah. But yes, I'll follow up with you on the other suggestion. John had one.
So Brian, how do I think you showed us some pretty compelling data for lactylation and its binding by TRIM33, I think it was.
Mm-hmm.
But there's a lot of literature out there right now that suggests there's a lot of claims for lactylation controlling everything.
Mm-hmm.
And you know, as a chemical biologist and interested in post-translational modifications, could you just give us your sense, a reality check about, you know, yet another histone or protein acylation modification, and just maybe I'd like to hear your opinion about, you know, how much of this should we believe generally as a major post-translational modification, or is it very specific to, you know, a very unique metabolic condition, a very unique set of proteins that actually recognize and do something with that particular mark?
Yeah, certainly since we started this project, there's been so many lactylation papers that you can't keep track of. And I think it's partially, I don't know if the antibodies are good, or at least they give you data. And so I think a lot of that can probably be taken with somewhat of a grain of salt. We're trying to give it some sort of mechanism other than just lactylation happens, gene transcription changes, and we don't know why. We think this might be one way, but there's been a few other lactylation reader papers that come out recently as well. So we'd like to look at that compared to ours and see which one's stronger. I mean, I will say, like, inherent to our study is we can bind lactylation, but the affinity for lactylation versus acetylation actually not that different, which maybe that's important. Maybe one way to think about it would be, you know, acetylation can happen everywhere. A lot of brominates combine acetylation, where lactylation happens. Only TRIM33 can bind that, and it's more of an exclusionary principle. So it just excludes other bromonames from binding there, even though there's overlapping specificity for acetylation. I think that's maybe one way that I'm thinking about how you might get selectivity out of essentially a chaos of different modifications. If only certain ones combine to certain ones, then maybe that helps partition where they actually bind on chromatin. But yeah, as far as the overall field, I can't imagine lactylation is doing as many things as people say it is. I don't know if anyone, maybe you know, I don't know if anyone's really quantitated the levels of lactylation versus acetylation.
I was hoping you would do that, Brian.
Well, yes, that's the next thing. I mean, Yingming Zhao did a nice Nature Chem Bio paper where he showed there's two diastereomers you can get when you do lactylation, because there's a stereocenter in it, and that it's the one derived from lactate rather than the one derived from, well, there's another pathway, or glyoxylase. You can also get lactylation, but that's the opposite stereoisomer, and it seems to be almost entirely, or just entirely, the one that's derived from lactate in the sense from glycolysis. So, you know, that was an important paper, I think. Thankfully, we picked the right one. We had to make a decision at some point which isomer we were going to screen. I mean, we took an educated guess. I don't know if that answered your question, but that's kind of how I'm thinking about it.
Thanks, Brian.
Okay, well, why don't we thank Brian again for a really exciting talk?